Chronic diseases, are they your fate? Today we want to address whether they're predetermined or whether there's something we can do about them. And the World Health Organization identifies these chronic diseases, the top drivers of the disease burden globally, as cardiovascular disease, cancers in its various forms, respiratory conditions, and diabetes. No doubt we know of someone in our family, our friends, our circle of influence that have experienced one or maybe multiples of these conditions. And particularly when we talk about families, it's something that we inherit, that's part of our nature, or maybe even something of how we've been brought up, our nurture, that's going to determine whether we're susceptible to these or not. Certainly geneticists would tell us that it's our nature, that we simply inherit genetic dispositions that make us susceptible to these diseases. Certainly, they've identified over 900 distinct disease genes. And within these, they've classified them into functional genes, and they're certainly functional in terms of how the disease gene is expressed within our body, I suppose we could say, and how it contributes to not only the type of disease, but also the age in which that disease is likely to affect us. So the first classification is that of transcription factors. Now, transcription factors are proteins that control the rate of the copying of genetic information. And essentially their function is to turn on and turn off genes in order to make sure that the genes are expressed in the right cell at the right time and in the right amount. And typically, these transcription factors, if that's the driver of the disease, is going to appear in utero. The second classification of functional disease in terms of functional genes, rather, in terms of disease onset and progression, is enzymatic. And these can be either defective or simply functioning in a reduced capacity. And the clearest example would be that, I'd say, of albinism, where there is an inability to process melanin. And almost a full third of the 900 or so identified disease genes fall into this category. And this is the conditions that typically appear in the first year of life. The third classification is that of receptors. Receptor genes are essentially a class of protein that, when activated, work on a secondary protein, and these control the development and metabolism, and typically appear in diseases that manifest themselves from the first year of life through to puberty. And the final category that they've identified is protein function modifiers, and these are the ones that regulate specific protein activity, and they account for nearly 15% of disease genes, of those 900 or so that have been identified. And they include proteins which stabilize, activate, or otherwise influence the secondary proteins. And these are the diseases that typically appear during adulthood. The researchers noted that with the abundance of enzymes in the disease gene set, may reflect some historical bias towards metabolic disorders in the study of human inherited diseases. And the argument is that it is in the first year of life that we establish our own homeostasis rather than that from our mothers, as we transition from placenta-fed, breastfed to more independent feeding and living. This acknowledgement of heritability is a reflection of the understanding of our genotype-phenotype mapping, where the genotype represents our full inheritability and our phenotype the observable or expressed inheritability. Genomic variations such as mutations and duplications can influence our phenotypes. However, studies into identical twins reveals that our phenotypes aren't only expressed based on our DNA. With identical twins, their DNA, their genome, is the same, but they display differing phenotypes. And we can see here on the screen the difference between 3-year-old twins and 50-year-old twins. Now, obviously, the 3-year-olds have been in a very stable environment. They share that environment, and we can see much of the same expression in the epigenetic tagging as opposed to the 50-year-olds, where clearly there is greater differences. And perhaps the easiest and most observable in the differences of phenotypes is in the fingerprints between identical twins. So what is it that contributes to this phenotypic variation or plasticity? And it's more than genomic, it is epigenomic, and that's why we're looking at epigenetic tags. By definition, epi meaning above or acting upon the genetic code. So there's not something that we're programmed for, as geneticists may suggest, that it's all in our genome, that there is in fact epigenetically that will feature in our nurturing, or we might say in our environmental influences that determine which genes are switched on and which are switched off. And these processes of epigenetic nature have been identified as DNA methylation, histone modification, chromatin modification, and non-coding RNA duplication. Without wanting to give the impression that only one mechanism is operable at any given time, for in fact, many of the mechanisms are acting simultaneously or concurrently, I'm really only going to talk about DNA methylation due to our time constraints, for it is implicated in each of the various nurturing factors. And we can measure all these in various bodily tissues depending on the specific outcomes that we are looking for. But as we're talking about nurture, that may be maternal, it may be familial, or it could be our extended social. Each has a role to play in our behaviour development and indeed our behaviour expression, even influencing our adopted values, our sense of identity. A secondary consideration in nurture or our environment is our diet, and this can include or exclude vital nutritional compounds that are necessary for DNA methylation. Our sleeping habits can influence not only our ability to make sound judgment decisions, but impact total neural activity, our synaptic plasticity, and our susceptibility to mental illnesses. And finally, perhaps what we might more think of our environment as the encountered pollutants through daily living. These have a cumulative effect on our bodies and can lower our methylation levels. On a global level, bodily, it is desirable to have high methylation levels. And the big deal about DNA methylation and the contributors to promoting or detracting from healthy methylation is that DNA methylation works directly in the copying of genetic information to express, unlike the disease genes, which work indirectly or secondarily. Further epigenetic expressions are generationally transferable. The Surgeon General suggests that even up to 5 generations could be impacting your health today, and then obviously the health that you then pass on to future generations. So, can we in fact dodge the chronic disease bullet? Yes, we can. As we understand these epigenetic switches, we have a powerful way of influencing our own susceptibility and vulnerability, and then also influencing that for our children, our grandchildren, and moving forward. So, what are those? Diet. We want a diet particularly rich in B2. So, we're thinking mushrooms and spinach, almonds. We want a diet rich in B6, pinto beans, pistachios, avocado. We want a diet rich in B12, nutritional yeast, fortified plant-based milks and cereals. We want a diet rich in folate. So we're thinking chickpeas, lentils, spinach, avocado, broccoli, along with a diet rich in lycopene, tomatoes, watermelon, papaya, carrots, mango. We want to include soy in our diet. We want to even consider some green tea. Why? Because these hold the elements that are essential for high levels of global methylation in our bodies. We want to get sleep, good sleep. We've probably heard it before, but most adults will need at least 7 hours of sleep per night, as recommended by the Sleep Health Foundation. We have an increasing developing understanding of the influence of circadian rhythms on total health, and this would indicate to harmonize the natural rhythm of our bodies and sleep according to our body's natural timing. We want to consider exercise. In addition to heart health and healthy weight consideration, exercise induces positive effects in both muscle and adipose tissue that contributes to healthy global methylation levels, and these similarly promote healthier weight. And of course, we want to have environmental management, and this includes positive relationships, parents, siblings, friends, so that we avoid feelings of isolation and thereby have effective stress coping mechanisms and supports. It also includes managing the environment that we work, rest, or play in to minimize toxins either inhaled, ingested, or absorbed. The way moving forward is that we have powerful control over our own susceptibility to chronic disease as well as future generations.